Okay, well, I'll start just by thanking the organisers for inviting me to this great meeting and this beautiful city. It's my first trip to Sevilla, and it's stunning. It's been a very enjoyable meeting so far. Today I'm going to be talking about work that's been going on in my lab and in very close collaboration with Josephine Pemberton's lab over the last 10 years or so on Soay sheep. So the work I'm going to be presenting has mostly been done by a couple of postdocs, Jake Grattan and Felina Fauna, and a PhD student in my lab, Susan Johnston, and in very close collaboration with Josephine Pemberton, who not only is a long-term collaborator, but she is the person more than anyone that is responsible for this long-term life history, individual-based study, being a great ongoing piece of work for 20-odd years. So I'm going to start just by describing the study system, and in particular 3 single locus polymorphisms that segregate in our study population, which are going to be the focus of the talk today. I'll briefly describe how we discovered the genes responsible and how we've shown selection at these loci, but most of the talk is all about the origins of this polymorphism that I'm going to talk about, and also trying to distinguish whether changes in gene frequency that we see are happening because of microevolutionary processes or because of genetic drift. So the Soay sheep project. Soay sheep are found on a couple of islands off the northwest coast of Scotland and in the St Kilda archipelago, which is at the very far north of the Mediterranean, right on the edge of the Mediterranean, up here. And Soay sheep have been the focus of a long-term study since 1985. Soay sheep are the most primitive breed of sheep found anywhere in the world. They're thought to have been on these islands for 4,000 years. They very closely resemble, we believe, the first sheep that were domesticated. So they massively predate the agricultural revolution. They're really not very similar to modern domestic sheep. Since this study's been going, about 8,000 sheep have been tracked and followed throughout their lifetime. So all of the sheep born in our study area are tagged as lambs, usually when they're between 1 day and 2 weeks old. We follow them throughout their lifetime. We collect a blood sample when they're born. They're caught many times over their lifetime, and usually we find out when they die as well. We find the bodies when they die. They're completely unmanaged. So there are very extensive data on longevity, lots of different life history traits, morphological traits, reproductive success, parasite loads, etc., etc. One of the most interesting things about Soay sheep, one of the reasons why ecologists first started to study them, is their population dynamics. So because St Kilda and Herta, the island we study them on, is quite small, they quite regularly reach carrying capacity and then have population crashes. So this just shows the population size in our village-based study area over the last 20 years or so. And you can see that these very heavy episodes of mortality happen quite frequently. It's not cyclical, but it happens frequently. It's very male-biased mortality as well. The mortality nearly all happens during the winter after the rut has taken place. Okay, I mentioned briefly there are some polymorphisms in the population. The 3 that I'm going to talk about today all behave as single locus Mendelian traits. The first one is a coat colour polymorphism. About 3/4 of sheep have dark coats like this, and about 1/4 have a light coat. It's not white, as we see in domestic sheep, it's a light brown. There's also a coat pattern polymorphism. This is completely independent. It's caused by a different gene on a different chromosome. And the majority of sheep have what we call the wild type coat pattern, which is characterized by this lighter wool on the belly, around the rump, and on parts of the face. This other pattern, which is uniform all over, is known as the self-type. About 5% of sheep have the self-coat pattern. We know the genes, and in fact we know the causal mutations that underlie these particular polymorphisms. And the third polymorphism is a polymorphism in horn type. Here we know the causal gene. We don't actually know the causal mutation, but we have a SNP that tags it very well and predicts phenotype very well. Briefly, males are either normal horned, like this guy, or occasionally they have much smaller horns. Sometimes they barely break the skin, known as skers. And females can either have normal horns, which are smaller than male horns, they can have skers, or they can completely lack horns. This is known as polled. And it's rather sad to summarize 5 years of work by quite a lot of people in one slide, but here I am. So the underlying loci are TYRP1 for coat color. There's a non-synonymous substitution, which results in where the dark associated allele is dominant to the light one. The coat pattern one is caused by the agouti signaling protein, which you heard about in Alex Ruan's talk yesterday on the owls. There's actually 3 different mutations in this gene, which is relevant to the story later on. One of them is a non-synonymous substitution. Another one is actually a frame shift substitution. And we are pretty sure there is a third regulatory substitution, although we haven't actually discovered that one yet. They're all in very strong linkage disequilibrium, so they essentially behave as one single allele. So just think of that as the non-functional version of the agouti signaling protein. And the horns polymorphism is caused by a gene called RXFP2. We don't know for certain the underlying mutation. It's probably a large insertion, and it appears that the same gene and probably the same mutation explains the absence of horns in most domestic sheep. These 3 different loci are all under selection in our population. One of the nice things about having 25 years of data, where you've got very extensive records on reproductive success and survival, is that you can identify fitness effects at individual loci. So with the coat color gene, TYRP1, what we see is that males who are homozygous for the dark coat color are less fit, sorry, not just males, this is in both sexes in this case, individuals who are homozygous for the G allele, the dark allele, are less fit than heterozygotes or light-coated individuals. At the agouti signaling protein, it's slightly more complex. Again, the effect is in both sexes, but the fitness effects vary across different life history stages. So in juveniles, individuals with 2 copies of the self allele, the non-functional version of agouti, have lower juvenile survival, but that's offset by, in adulthood, they have higher reproductive success. And in the case of horns, there's no fitness consequences in females, but in males we have a trade-off between survival and reproduction. And what we actually see here is that plus males, who are the males with the longest horns, have intermediate fitness because they have very good reproductive success, but they have relatively poor survival. PP males tend to live much longer, but their annual reproductive success is quite low, and heterozygotes have reasonably high reproductive success, reasonably high survival, and overall a higher fitness. So this trade-off between natural selection and sexual selection results in the polymorphism being maintained. So one of the things we've been interested in is, having shown that these individual genes are under selection in our population, we've been looking at changes in allele frequency over the 25 years that we've been studying them. And because we can type the causal mutations, we can detect changes in allele frequency. One of the questions one would want to know is, if you see a change in allele frequency, is this a microevolutionary response to selection, or is this just dynamics due to genetic drift? Remember, this is an island population. The effective population size is actually not that small. It's around about 200. But nonetheless, you still expect drift to play a role. So I'm not going to show these kinds of plots for all of 3 polymorphisms. I'm just going to focus here on the coat pattern one. So this plot shows the frequency of the self-coat phenotype over the 20 or 25 years or so of our study. And what you can see is that it's actually declined in frequency, the self-coat pattern. That's this one here. And if you do a regression of frequency against time, you get a significant slope. If you run a mixed model, which corrects for some kind of autocorrelation, it's still statistically significant. And of course, we know that this locus is under selection. What we want to do, though, is try and distinguish whether these changes can have to have happened because of selection, or whether similar changes could have happened by drift. And one of the ways we can do that is by taking advantage of the fact that we've got this very detailed pedigree going back 20 years or so. So what we can actually do is, because for most individuals we know who both parents are, sometimes you only know who one parent is, we can essentially do what we call a gene dropping exercise. So we can take our pedigree and we can simulate alleles that match our allele frequencies at our causal loci, and we can make them segregate down our pedigree in a completely random way to simulate drift. So imagine our founder individuals, let's call them generation 0. What we're going to do is we're just going to assign alleles to those different founders using the frequencies that we found at the start of the study. And what you can then do is you can make their offspring inherit one or other allele from each parent at random. So this just simulates genetic drift. Occasionally, individuals further down the pedigree won't have both parents known, in which case you just assign them an allele that's representative of the allele frequencies in the cohort in the year before they were born. And we repeat this process until everyone in the pedigree has been assigned a genotype. And what we can then do is we can perform a regression of allele frequency against time. And if you do this lots and lots of times, 10,000 times say, then you generate a distribution of what you expect your regression slopes to be just due to genetic drift. So this was that same plot I showed of the change in the coat pattern frequency. And what you can see here is that although that was a significant slope, drift can cause similarly significant slopes by chance very often. This is our distribution of expected regression slopes under genetic drift. And you can see that our dotted line here, which is our observed value, is kind of pretty much slap bang in the middle of the distribution. So although this gene is under the gene underlying the polymorphism is under selection, you could get those changes just due to drift. What's actually quite intriguing is if you look at the actual allele, the self-allele, rather than decline in frequency, it's actually increased in frequency. So remember, to have the self-phenotype, you have to have 2 copies of this allele. It's a recessive allele. So the actual underlying, although the phenotype has declined in frequency, the allele that causes it has increased in frequency. But again, it's still no different to what we expect from drift. What is quite intriguing, though, is that the number of heterozygotes in the population has gone up. And it's gone up significantly more than you expect by drift. And similarly, if we look at the ratio of the observed number of heterozygotes to the expected number of heterozygotes, it's gone up more than you expect just from genetic drift. So here we've got a pattern that can't just be explained by genetic drift. And I'm just going to have to go back. I meant to put an extra slide in. Sorry about this. Probably what's going on here, I suspect this is an ephemeral trend that we're seeing, and I think things will stabilize to something closer to Hardy-Weinberg equilibrium. But if you imagine selection or viability selection against these individuals, that explains why the self-phenotype has gone down. But because the ones that survive have high reproductive success, and they are relatively rare, most of their matings will be with either homozygotes or heterozygotes rather than with other self-homozygotes. So most of their offspring will probably be, or a high proportion of their offspring, will be heterozygous themselves. So I think that's why you see this increase in the frequency of the self-allele, at least ephemerally, and therefore an increase in heterozygotes in the population. For the purposes of this talk, I think that the main message here is that actually it's quite hard, even for loci under selection, to be able to distinguish whether microevolutionary trends have arisen due to selection or drift. The selection coefficients here are actually quite strong, and the ratio of the product of NE and S is much greater than 1, which is relevant for whether alleles are evolving due to selection or drift. So it's actually, although the gene dropping method is quite an attractive way of trying to distinguish whether trends are due to selection or drift, it's very easy to get quite dramatic trends just from drift, and it's very hard to demonstrate that trends are because of selection rather than drift, even at loci that you know are under selection. Okay, the second part of the talk is looking at the question of where these polymorphisms come from and whether we have evidence of domesticated alleles being introgressed back into a wild population. I haven't really spoken much about the history of St Kilda or the human history of St Kilda yet, but St Kilda is actually 4 islands, only one of which has ever been inhabited by humans. It was inhabited by humans until 1930, and they mainly survived by harvesting the seabirds on the precipitous cliffs there. But they did do some farming of livestock. They kept some cows, and they kept sheep. You can see some of them with sheep. in this figure here. And what you'll notice is these aren't Soay sheep. These are a now extinct breed called the Old Scottish Short Wool or the dun-faced sheep, which they kept until about the mid-1800s. And you may have read a recent paper by Phil Hedrick, which has just appeared in Molecular Ecology, and it's very relevant to the story I'm about to tell. It's looking at the question of whether adaptive integration has been happening in animals, and in particular the focus of that particular review, and it's a meta-analysis as well, is looking at the question of whether adaptive integration happens quite often in mammals, so genes being introgressed, particularly from domestic populations into wild ones, and perhaps the focus of that particular review is that can give quite different evolutionary dynamics than the classical explanations of how adaptive polymorphisms have arisen, which the classical explanations are either de novo mutations or standing variation that eventually becomes adaptively important following, for example, an environmental change. So we got interested in the question of admixture, partly because we knew there had been other sheep breeds on St Kilda, but also when we first started looking at some of our population genetic data, we saw very long-range linkage disequilibrium around some of the genes we were interested in, which can be a consequence of selective sweeps, but similar patterns can arise when admixture has happened, and it got us wondering about admixture. And then we discovered a paper written in 1912 which described how the St Kildans had put some of their sheep onto Soay. And I'll just explain this map quickly. Our study area is on the main island Herta, which is the island where the St Kildans lived. Soay sheep until 1932 were only on the island of Soay, where they're thought to have been for 4,000 years. Soay actually means it's Norse for island of sheep. So when the first Vikings arrived, there were already sheep on Soay. And in 1932, the Herta population was founded by moving just over 100 sheep from Soay onto Herta. So the St Kildans kept their sheep on Herta simultaneous to Soay sheep being on Soay. And this chap Ferguson, who was the postmaster on St Kilda in the mid-1800s, he didn't speak any English, he only spoke Gaelic, but what he said was translated, and he described how a few rams of the race which preceded the introduction of black-faced rams were once introduced into Soay, but they did no good. In other words, he's saying they moved some sheep onto Soay, but those sheep didn't reproduce. The Old Scottish Short Wool, the breed he refers to, or the dun-faced sheep, is now extinct. It probably went extinct in the late 1800s, but it probably quite closely resembles some of the sheep breeds found on some of the other northern Scottish islands, such as on the Orkneys. This is a Ronaldsay sheep. Ronaldsay sheep are quite interesting, and they have quite a lot of variation in coat colour. What happened is the St Kildans kept those until the late 1800s, and in most of Scotland, the Scottish blackface became the predominant breed. It's still the most predominant breed in the UK. So in the late 1800s, the St Kildans replaced their Old Scottish Short Wool or dun-faced sheep with Scottish blackface sheep. But they did hybridise the two to create something called the Borroray sheep. Borroray is one of the other islands on the St Kilda group. It's now a very rare Scottish sheep breed. It's only found in the wild on the island of Borroray. And Borrorays are relevant because we can think of them as a living museum in the sense that they carry Scottish blackface genes, but they also carry genes from this now extinct dun-faced breed, the breed that may have hybridised with Soay sheep. So this briefly summarises what's gone on. Soays have always been on Soay and on Herta since 1932. The dun-face no longer exist, but they're on Herta from probably about the 1700s to the mid-1800s. Think of them as a post-agricultural revolution breed. So they are still a modern breed, despite being extinct. Scottish blackface are now all over the place. They were introduced to Herta, where they were crossed with the dun-face to create the Borroray. This is the island of Borroray here. Now, we're very lucky in that by working on a breed that's of agricultural importance, there's quite good genomic resources that have been around actually for quite a while now. So we have a 50K SNP chip, so a chip with 50,000 SNPs on it, evenly spread across the sheep genome. At the time we did this work, about 500 of our sheep have been typed on it. It's now the case that all 7,000 sheep for which there are DNA archives have been typed on this chip. And usefully for us, 74 other breeds and various wild sheep species have been typed at exactly the same markers by the International Sheep Genomics Consortium, and we have access to all of those data. And indeed, we've contributed data towards that project. So what we set out to do was test whether there could have been admixture between these extinct but nonetheless modern dun-faced sheep and Soay sheep sometime in the 1800s, as Ferguson described. And what we did is we wanted to simulate a situation with and without admixture and see whether these SNPs that we've typed could have detected this admixture event that would have happened as a one-off event perhaps 150 years ago. So our null model is that there's been no admixture between Soay sheep and dun-faced sheep. I'll put a line through dun-faced just because that means we haven't sampled them. We've only sampled the Borere sheep. Remember, dun-faced are extinct. As far as we know, there are no museum specimens of dun-faced sheep. And the breeds we've looked at on the SNP chip are the breeds which have been on St Kilda, and as an outgroup, we've chosen an African breed as well, the red Maasai, which presumably have evolved for quite a few thousand years independently of these Scottish breeds. And then our admixture scenario is exactly the same, except admixture happened between Soay sheep and dun-faced sheep about 32 generations ago in our simulations, which is about when Ferguson described this happening. But our Soay sheep have been sampled subsequent to this admixture event. So what we can do is we can take our SNPs on this SNP chip, and we don't use all 50,000 SNPs. We prune out the ones which are in strong linkage disequilibrium with one another, because it's known that that can give false patterns that look like admixture. And what we do is we use the program structure to see whether we would detect admixture if it happened 150 years ago. And briefly, the different colours represent different genetic clusters, and what you can see is that most of the breeds come out as good clusters. Borere sheep come out as two colours. The pink represents their Scottish blackface ancestry. The dark green represents their dun-face ancestry. So when we simulate admixture between dun-face and Soays, you can see some dark green in the Soays. What you predict to find is that all of the Soay sheep that we sampled today should show some dun-face ancestry of roughly equal proportions. In our no admixture simulation, Soays come up as just a single light green cluster. And what happens is when we actually look at the real data, we get a very similar pattern to our admixture scenario. What you'll see is that Soay sheep have this dark green colour in a reasonably uniform way, and that the dark green is found in Borere sheep, but not in Scottish blackface sheep, indicating that it's this bit of dun-face ancestry. The other thing it shows actually is that Borere sheep have got a lot more dun-face than perhaps we expected. The influence of Scottish blackface sheep in the Borere breed is less than you may have predicted. So this looks like quite compelling evidence that dun-faced sheep contributed genetic variation to Soay sheep around about 40 generations ago. Or to put it another way, we now have a 150-year-long experiment where you've got domesticated alleles playing off against the more ancestral alleles in an unmanaged population that's under quite strong selection. And briefly, what we think has happened can be summarised here. From the Middle Ages until the mid-1800s, you had dun-faced sheep on Herta, Soay sheep on Soay. This was the admixture event. Then the Scottish blackface sheep was introduced to Herta and mixed with the dun-faced breed. In 1930, when the humans left, all the sheep were removed from Herta. In 1932, when our population, the slave population, was founded, we've got a mixture of light green and dark green. In other words, our mixture of Soay sheep and dun-faced sheep. And that's the population that we study today. So I guess the big question is, do our polymorphisms only occur in our population because of this admixture event? And it's interesting that the mouflon sheep, which is thought to be the ancestor of all domestic sheep, very closely resembles our dark, wild, normal-horned Soay sheep. So there's a mouflon, there's some Soay sheep with the dark, wild coat patterns and nice big horns. And the way we've addressed this question is by looking at something called haplotype sharing around the causal loci, the causal genes that explain our polymorphisms. So the idea here is that if two populations have quite a recent common ancestry, then recombination has not had much opportunity to break down associations at linked sites. So if you look at particular alleles as you walk along a chromosome, if two populations have a recent common ancestry, you should get what we call long haplotypes or haploblocks. And the longer they are, the more recent their ancestry. And what we can do is we can compare our Soay sheep with all of the other sheep that have been typed on this SNP chip and look at so-called haplotype sharing between Soay Soas and the other breeds around the genes that we're interested in. So to give an example of this, this is our coat colour gene, TYRP1. And what we do is, what we've got here is a table which shows the 6 SNPs closest to our causal variant. So there's 3 upstream and 3 downstream. And the numbers on the top are just the distance away from the causal variant. So if we look in our Soa sheep, we can see that there are 7 what we call core haplotypes that we find. And 5 of those associated with the dark coat colour. In other words, when you find that haplotype, it's nearly always with the dark allele, and 2 of them are with the light coat colour. And this row I've highlighted here, this is the most common light haplotype. So most of the light sheep have this particular haplotype. So what we can do is we can take these core haplotypes and we can ask whether you find the same haplotypes in other breeds, and if so, how long the haplotypes are. And by that what I mean is we walk away from the core haplotypes further and further away to SNPs further and further away, and if the Soa haplotype and the thing you're comparing it to has the same allele, then you're extending the haplotype. So for example here, they both have a G. If you look at the next site, another 200 kilobases away, they both have an A. But then eventually when we get to this site here, they have different alleles. So our haplotype, our shared haplotype is this along here. So we can do that for every pairwise combination of core haplotypes and the different breeds, and work out a mean haplotype sharing of each haplotype between Soas and between each of the other breeds on the hap map. This rather noisy slide summarizes that. I'm going to zoom in on the relevant bit in a minute, but just to point out what's going on here. On the y-axis, we have mean haplotype sharing, and the letters just refer to the different breeds that we're comparing Soas to, and the different colors represent the different haplotypes. So if we zoom in, so just going back, what you'll notice, perhaps the most striking thing here is that these breeds here have much more haplotype sharing with Soas than any of the other breeds. And if we zoom in on them, these are our Borere sheep. Our Borere sheep come from 2 places. Some of them actually come from the Isle of Borere. It's very hard to get samples from there. Some of them come from a mainland population which is maintained by the Borere Breed Society. But because it's a mainland population that's been on farms with other sheep breeds, there's always a risk of some contamination. The light blue bar here represents our most common light haplotype in Soa sheep. And what you'll see is that this light blue bar has very high or long haplotype sharing between Soa sheep and Borere sheep, much longer than we see for any other breed with Soas, and also much longer than we see for the common dark haplotype, which is this dark brown one here. And this is a significantly longer haplotype sharing between Soas and Boreres than between Soas and any other breed. So this suggests not only that the light haplotype is found in both breeds, that it's been in both breeds relatively recently, and much more recently than any other breed that we can compare Soas to. And we can do a very similar analysis on the agouti gene, the coat pattern gene, and show that the self-coat pattern shares a similar recent origin between Soas and Boreres. Interestingly, when it comes to horns, the polled gene, the polled allele, the lack of horns, or the short horns in males allele, doesn't seem to show any particularly strong haplotype sharing between Boreres and Soas. So the suggestion is that the light coat colour and the self-coat pattern have recently been introduced to the population through this admixture event, but the horns polymorphism may actually be much older. And let's just recap the fitness effects that I referred to earlier. So we can see here that the T allele at TYRP1, the coat colour gene, this is our introduced gene, and we can see that actually the least fit sheep are the ones that are homozygous for the ancestral allele. And similarly at coat pattern, the non-functional version of agouti, the self allele, is the introduced one. It's associated with reduced juvenile survival, but it's also associated with increased adult reproductive success. So what we can see here is that these domesticated alleles are not necessarily associated with lower fitness when they're reintroduced into the wild. We don't know whether these alleles represent standing variation that was around in the ancestors of all sheep at low frequency, or whether they're alleles that arose de novo post-domestication and were then increased in frequency due to artificial selection. We certainly know they've been increased in frequency due to artificial selection, but we don't know exactly when they arose, whether they arose before sheep were domesticated or not. But in the case of Soa sheep, we can be pretty sure that they've only been around for about 150 years ago, and that they don't appear to be associated with any great fitness cost. So to conclude, the gene dropping approach is something that can be applied potentially to any population where good pedigree data exists. It's a promising approach for detecting microevolutionary trends, but it is quite hard to distinguish drift from selection. It really does need quite strong selection to be sure of that, certainly in populations with reasonably modest NE. We can be pretty sure that admixture occurred between Soas and this more modern breed, probably happened about 150 years ago, and it's probably the direct cause of the two coat polymorphisms we see in the population, but possibly not, or probably not, the cause of the horns polymorphism. And that this 150-year-long natural experiment where domesticated genes have been in competition with ancestral genes suggests that the domesticated genes don't necessarily do badly in the wild. Maybe that's because they've been quite carefully, if they had been deleterious, they probably wouldn't have risen to high frequencies during artificial selection and domestication anyway. And with that, it leaves me to thank you and to take any questions.